So now we've heard the last presentation for today. This, the first of our 3-day journey exploring the frontiers of new scientific knowledge. It is through gatherings such as these, envisioned under the banner of the Molecular Frontiers Foundation, and partnering with some of the world's most prestigious universities, such as the Hong Kong University of Science and Technology, that creates the opportunity to bring together the brightest minds in fruitful discourse. The founding purpose behind Molecular Frontiers is to foster exploration and discovery, connecting scientists from a multitude of disciplines with young people around matters of global consequence, and in this way fostering new perspectives that can in turn lead to eventual breakthroughs. And we have every reason to expect that this symposium will achieve exactly this. Now, if I can invite all of the speakers from today to come up on to the stage, and also Professor Marquez, if you can come as this morning's chair. Please join me on the stage for the panel. So if you have a question, if you could just please raise your hand and someone will bring you a microphone, and let us know your name and your affiliation. And this is your chance to ask your questions to the speakers. So hopefully, I can imagine that you have a lot of questions from today. We covered a wide range of topics. There's somebody in the back.
Hello. Thank you. Thank you for today's very interesting talks. So I'm from HKUST here, a PhD student majoring in life science, which we study the developmental biology and the stem cell, probably cellular therapy, which is different from the maybe Dr. Zhang Feng's work, like gene therapy. So I kind of have two questions regarding Dr. Zhang Feng's work. So maybe first is about the PNMA2. So you show that it is a potential tool for drug or for the mRNA delivery. But I was wondering, the PNMA2 actually is shown it's involved in the autoimmune system response, and also PNMA2 will cause the neuronal disorder, right? Which is a paper published just 1 month before your PNAS paper in the Cell. They show that if you inject the PNMA2 to the mouse brain, it will affect the mouse, cause a neuronal disorder. And second question, this is the first question, so whether you also observe some side effects. If you observe side effects, how you solve this? And the second question is that in your, you try to engineer to deliver 3 factors, try to recover the aging immune system based on my understanding. So I was wondering whether these three factors actually you saw is really kind of recover the aging immune system, but I was wondering whether what's really behind, like whether the cellular changes, a more immune cell active, mature active immune cell were produced or not. Yeah, that's my questions.
Great, thank you so much for the questions. These are excellent. So for your first question, with regard to PNMA2,
Yeah.
so there are two aspects to the PNMA2 protein. There is the capsid domain, which allows the protein to aggregate or to oligomerize and form the capsid. And then there's also an N-terminus spike domain that forms nodules on the outside surface. So there are several ways to engineer PNMA2 to remove some of the interactions that you mentioned. And a lot of that involves modifying the N-terminal spike domain. In fact, you can cut it off and then replace with other spike domains to be able to change targeting specificity or just interaction with other proteins in the human body. So that's how sort of I would go about engineering the system further. And then you can also engineer that domain for either cell type specific targeting, or you can engineer for endosomal escape. So by changing the activity of that domain. As for the second question, with the three factors for immune rejuvenation, one way to measure what is happening to the T cells is by measuring the diversity of T cell receptors. Oh. So what happens over the course of aging is that your T cell receptor diversity declines, and specific subsets of T cell receptors, they become overpopulated, overrepresented in the body. Mm-hmm. So when we give these factors, we find that there's a more even distribution of T cell receptors, and there's more diversity in the T cell receptor, which is more resembling a younger immune system. So that I think is kind of the key mechanism.
Oh, okay, got it. Thank you.
Thank you.
Is there another question? Oh, right there in the few rows down from the last question. There's a right here, a woman with her hand up.
Hi, this is Xiaoying. I'm with a GF Investment. I'm in the sector of private equity and venture capital investment. So it's really great pleasure to have all of your super high-level scholar here in Hong Kong. So it's really, really our great honor to have you here. So there's just one general question I have to ask is, we like to learn your past experience in interacting with the Hong Kong science community and your future plan or vision of how to interact with the Hong Kong science community. We definitely love to hear each of your sharing. Thank you.
Should we start right here?
Oh. Well, actually, this is my first visit to Hong Kong. And so far, I have no interaction with the people. That means the hope. And I hope I can do a lot here because I know now the standard of science and technology is really high here. And I so many enthusiasm here. So I love to do some collaboration in Hong Kong in near future. Thank you.
Ditto. It would be great to interact.
Well, this is my third visit. I got an honorary degree here, but also one of my students was on faculty here, or actually a couple of my students are at different faculty positions here. And then one of my students started a company and asked me to be an advisor to it, so I'm doing that too. In fact, seeing him tomorrow, hopefully to see what's going on.
Almost likewise, several of my ex-students are now professors here, and I shall be seeing them next week and continuing.
So I'm
involved in the Croucher Foundation. So I'm now the chairman of the Croucher Foundation, which is a Hong Kong organization which supports young people, in particular, going abroad, and also bringing some exciting science to Hong Kong. So hopefully unless they depose me, I shall continue to do that.
Yeah, it's my third or fourth visit in Hong Kong, but I haven't been here for quite some time. And when I got here, actually yesterday, and I had a visit at another university this morning, I could witness that Hong Kong has, of course, thrived since then, and it has become a very vibrant place. I was also surprised to see that some of the methods that I'm developing are being taken up at very fast pace. I didn't anticipate that. And so I very much look forward to interacting with the Hong Kong science community, and and I look forward to seeing Hong Kong develop further and getting ahead very quickly.
This is my first visit to Hong Kong, and it's been wonderful so far. Clearly that you don't have the same opioid crisis here as we have in the U.S., but I'm eager to learn more about, I'm sure there are addiction problems here, and also HIV and correctional health care. So happy to continue.
This is my second time in Hong Kong. The first time I was here in 2018 for the second International Gene Editing Summit. And there I sort of interacted with the different sort of scientific communities here. I'm also a member of the Hong Kong Life Science Advisory Board, which engages in life science education for, I think, high school and also college students. So I think it's a great community here.
Thank you so much. And as we are a very active investor in the science field, we have many outstanding portfolio, including Twist Bioscience in the US and UltraDX, which is also a sponsor of today's event. So we look forward to have the pleasure and honor to interact with many of your very honored professor. Thank you for coming.
Thank you. There's a question right a little bit down the same aisle, right there.
My first question is addressing to Professor Rob Langer, because I think your speech is the most engaging one today for a general audience like me. My name is Shu Yan, graduate from U of T. You are a chemical engineer, you are a biologist, but you're also a businessman. My question is, how do you actually turn the science into business? And how do you actually monetize the value of the science in the biotechnology field? And then what's your exit strategy when the Moderna stock price has been dropped from the $484 to $40 as of yesterday, when your personal wealth dropped from 1.5 billion to maybe 450 million? So just curiosity.
Wow. I'm not sure where to start. First, I don't know that I'm either a biologist or a business person. But beyond that, I mean, nor do I have an exit strategy. I mean, I try to do things on the science side, and I work with people who are, who really do know business. And I think it's, I look to them to do business things, but I think I've been able to help on patents and the science and the scientific strategy. To me, any of these companies are a team effort. In terms of exit strategy, again, I think I don't really have one. I mean, the goal, in fact, even the investors that I work with, the exit strategy they say is, well, you just want to keep building value and building value. If somebody comes along and wants to acquire the company for, maybe a lot more than it's worth, they'd say, well, sure, we'll do that. That's, to, I suppose, maximize shareholder value. That's what I've learned. If you take a company, I mean, like you asked about Moderna, well, I mean, the stock's been all over the place over a number of years. It's been as low as 12, and as you said, as high as 400. I expect it'll continue to be very volatile. But I got involved in doing it because I wanted to see the work we did, make a difference. which I'd like to think that it has. And, so I mean, and believe me, when I started doing this stuff, I never dreamed that, I'd even come close to getting the wealth that you mentioned, so.
I also think that one of the points of Molecular Frontiers is to bring to audiences some of the cutting-edge scientists who are making discovery and breakthroughs that sometimes becomes something more than what they're doing in the lab. But all of these scientists today and all the ones that we've used at our other conferences, they enter into this field because of their passion. And sometimes it creates great wealth and other times it doesn't, but that doesn't change their love for what they're doing. And I think that even those that haven't become as successful monetarily feel just as wealthy. So that's one of the messages we try to convey to all those who attend these programs, that it's about the passion.
Thank you. And a follow-up question is for the general public. I would like to know if you have to predict what's the next biotechnology breakthrough in which field, and then we'll save maybe tons of lives and save tons of cost and maybe win the next Nobel Prize. Thank you.
I don't think that probably anyone here has that kind of information. It's done by committee over many, many years. Sometimes there's a breakthrough that's so obvious that maybe that gives you a sense, but I don't, maybe you could tell us.
Oh, no, I cannot tell. But one thing I can tell, that usually breakthroughs come from a direction that you don't expect. That's why it's a breakthrough. Breakthroughs are not really predictable. I think it's in its very nature. And so that's why I'm not venturing personally into this is going to be the next breakthrough. Well, by the time it is a breakthrough, it's apparent that it's a breakthrough. And then many people move in that field. And of course, that may be an important field, but solutions very often come from a direction that you do not really anticipate. That's very normal.
I think if you just look at the prizes from this year, 2 of them related to AI, and AI was developed so many years ago, and it's only now that it's being recognized in this way. But 40 years ago, when it started being formed, I don't think anyone would have thought back then that it would come to 2 Nobel Prizes. How about down here? And then you're next.
Thank you. Hi. First and foremost, I just want to thank all of you for the life-saving work you're doing. I'm going into computer science, but just hearing these talks makes me rethink all my life decisions. It's just, it's so incredibly fascinating and very, very deeply meaningful. So I'm from the States, and I hate to get political, but I get very fearful when I see, the propagation of misinformation. I see, our next health secretary is a vaccine denier, and I guess my questions, and perhaps directed especially to Professor Langer and Professor Rich, is how do you tackle these issues, both on the personal, familial level, but also in larger national discussions?
I'll take this soft one. I'm pretty ashamed of our the results of our national IQ test. We seem to have failed. This is uncharted territory in many ways, and I think it's usually we kind of think we switch from offense to defense to offense. That's the way our political system. But this has kind of gone off the rails a bit. I'm hoping it'll pull us back. But it's given me opportunity to kind of take stock of, you mentioned personal, like, okay, is my daughter going to be okay? Are we going to be okay? And then think, beyond that, okay, how's our state going to be? How's our city? And how, my patient's going to be? And how, just kind of thinking through in those terms, what are the implications? There are lots of places in the world, and probably most people live in places where the government is actually not helping to facilitate better lives for the people. But I think that's what we want to get back to. So the vaccines is a great example. I mean, we need to understand, how did all this vaccine hesitancy, how did that come about, and how did it become so, so mainstream? And I'm just thinking, Rhode Island, we're the smallest state, and we have a population of 1 million, and there's no reason we shouldn't have the highest vaccine rate in the country. So this is a great laboratory to actually try and understand, what works. We educate the parents, educate the school children, educate the teachers, and then seek out the people that are vocal opposed to vaccines, and try and have public discourse, and try and understand where they're coming from, and try and, I think, so there's things we can do, and we just have to focus on, what we can do, and double down on it.
I mean, I agree with that. First, he also hasn't been confirmed, but he may well be. I think we'll just have to see. But regardless of that, I think all you can do is what Jody said, and do the best you can do. And, I expect that what happened, I think this election, the pendulum swung a certain direction because of what happened the last 4 years. And if they do such a terrible job, I expect it'll swing back. But, I think you just have to do the best you can, and in terms of science, in terms of your lives. And I think obviously people will, if terrible decisions are made, I mean, there will be protests, and I think everybody will do the best they can to try to correct these things.
Thank you, professors.
Okay, this one right here. And I think there was a question in the back, was there? Yeah, okay. You're next.
Thank you guys for your insightful sharing. My name is Summer. I'm currently a final year student, final year undergraduate student from HKUST. So I have a couple of questions for Professor Stefan Hao and Dr. Zhang Feng. So my first question for Professor Hao is, have you ever experienced any challenges throughout your career? And if so, what advice would you give to students who are currently encountering or may encounter obstacles in their research? Yeah, and you can answer first.
I think most people who try to make some sort of big advancements or breakthroughs face challenges, but I think that is very common. I mean, initially I faced a challenge and no one believed that it's possible to go beyond the diffraction barrier, beyond the resolution barrier of a light microscope, because people had categorized light microscopy as something that is finite in its resolution. And then trying to think out of the box and convince people that this is not the ultimate limit for reason A, B, and C is very, very hard. And then I think one way of dealing with such problems is to really analyze the problem and check out if the reasons why people say it are fundamental, conceptual, or just technical. And if the concept is correct and the problems are just technical problems will be overcome with time. But the conceptual correctness, of course, will stay. And then it's easier to work towards that goal because you know that you're on the right track, the concept is right. So that helped me personally to persevere and to move on. And yeah, today we are basically down at nothing, at two molecules touching each other, still able to resolve them. And I think this is the only advice I can give, to really be self-critical and check if there is a fundamental conceptual problem. If there is none, just technical problems, time will sort out the problems. That's my personal view. I think personally, it's much more fun and much more interesting to work on problems that are challenging, problems that everyone thinks are not solvable, because of course the reward is high and the fun is high. At least that's how I see it. So I would be bored working in a field that basically everyone works on, because then competition is high and there's incremental advancements and people fight over small claims and this kind of stuff. And then that's not the type of science that I like personally. So that's the advice that I can give to you. But in general, I would say don't take advice. Do your own thing. Yeah, don't listen to me. Just do your own thing.
Thank you, Professor. So my second question is for Dr. Zhang. So as you reflect on your career, do you have like any moment that stand out most to you that is like very rewarding, and how has this like moments that has influenced your perspective on your career or your work in neuroscience?
Yeah, thank you for the question. I think there are definitely many moments that have had a formative role in my development as a scientist, as a person, all that. I think if I think about how I became a scientist, I think the most important part were the mentors who I met. So there were mentors who I met in middle school, in high school, in college, and in graduate school, and even after I started as an assistant professor to run my own research group. I think the most, probably, the person who had the most profound impact on my interest in science was probably the scientists who I worked with when I was in high school. I grew up in Des Moines, Iowa, and in Des Moines there was a research lab for gene therapy at one of the hospitals. And so when I was in high school, one of my high school science teachers came to me and he said, I found out that there's the new internship program that a hospital is having. You can go intern in any department. You can go into the operating room, you can go into internal medicine, or you can go into accounts receivable and just learn general vocational skills. But maybe you can also sign up and volunteer in this gene therapy lab. Maybe you have to wash some glassware and take out the garbage, but maybe you can also learn something about science. So when I joined the lab, I met my mentor. His name is John Levy. So Dr. Levy was one of the researchers working there to develop viral vectors for gene therapy. He came from a chemistry background. He was a PhD in chemistry, but he was inspired by what gene therapy and genetic medicine can do. And he was an incredible teacher. At the gene therapy lab, they had the Cambridge tradition where every afternoon at 4, people would gather in the lunchroom and have tea and have a cookie. John, he is very patient. He would just go there and everyone else would just come grab tea and then get a cookie and then take off, go back to work. John, he is a talker. He just likes to sit there and chat. And I was working with him, so I didn't really have anything else to do. If he was sitting there and chatting, I just had to sit there. And usually people would talk to him for about 5 or 10 minutes and they would take off. And I would sit there and he would talk to me. And in many ways, that was a huge benefit, a huge opportunity for me because he was extremely passionate in science. He would take out just blank sheets of paper and start to draw all sorts of fascinating things on there. He showed me what cocaine looks like. And then he also told me about green fluorescent protein and how the evolutionary history of jellyfish and how the biological life cycle jellyfish would play a role in what this protein is doing for it. And then he would draw, how retroviruses work, how all the genes work. And it's just the passion that he exuded and also the patience that he had with me to tell me about science that really kind of infected me with this enormous enthusiasm and excitement for science. I worked with him for 3 years in high school. So from 10th grade to 12th grade, I would go after school every day from 2 p.m. and stay until 7 p.m. and just work there, work with him. Many of the things that I teach my students today, I learned from John. And so that was one of the really important sort of factors that influenced my interest in biology. From that point on, I wanted to continue to do research. The second thing was also about John. So John was bipolar. He was affected by psychiatric illness. And so I never really sort of knew about psychiatric illness. And even when working with John, I didn't really fully understand what psychiatric illness was. I was under the basic understanding, or I was under the misconception that psychiatric disease is just people who are having a bad day. Everybody can have a bad day, you can recover from it. But having experienced some of that when working with John, and then in college, having really seen firsthand when one of my really good friends became sort of severely depressed, that experience got me excited, got me interested in working on neuroscience. And so that kind of led me to join the lab of Carl Deiserath to then start to develop tools for studying how brains work. So anyway, I think it's really the people, the experience, and the mentorship that really kind of guided me to pursue certain types of science.
So since we're on the topic of mentorship, which we know is a very important aspect of science and part of the reason behind these meetings, maybe we could just pass down the microphone and each one of you could talk a little bit about an important mentor in your scientific career, and then we'll get to the questions up in the corner.
Thank you. Thank you for your questions.
I've had many mentors along the way, and I've mentored a lot of people as well. And mentorship it can be a wonderful relationship, and ideally, it's a relationship of mutual respect. When I sign up to mentor somebody, I don't think of them as sort of junior in that they're any lesser than me. They're the same as me, but they're at a different point they have a different amount of experience and different training. And eventually they catch up, and sometimes they go past beyond me, and that gives me a lot of pride. So I think, but clearly, I didn't have as intense a relationship with a single mentor in high school, but I had several, and a few stand out in college, and then beyond. So I think that is really a critical part of science and medicine and public health. So,
thank you.
I had two very good physics teachers in high school, and I think I owe a lot to them because they kind of mentored me and encouraged me to study physics eventually. Later on, I was not that lucky, I must admit, but on the other hand, if I think in retrospect, in a sense it was luck, because this gave me this kind of independent way of thinking about problems. I think mentorship is great and it's important, but it should not be idealized, because sometimes it can also be a burden. So personally, I think if you don't have a mentor as such, don't worry. Just try to come up with your own view of what you want to do. So that's my personal take. While I'm trying to be a good mentor as well for my own students, partially because I didn't have my own mentor, I still think that they should become independent thinkers. And so please be courageous and come up with your own path.
I just want to add to that there are some really not good mentoring relationships. And those, people that want to just, they use you as cannon fodder. They work, free labor. And if it's not based on mutual respect, then move on.
Thank you. Professor Winter?
So I suppose I never thought of myself as formally having a mentor. It's only when I look back I realized that various people were effectively, in some ways, a mentor. In some cases, it's different things. It's really, it's the example they show. So my own formal supervisor, one of the great things I learned from him was his total optimism and enthusiasm about things, even things that were impossible. He could become very enthusiastic and very excited about them. And he would, there's a chap called Brian Hartley, who was a protein chemist. I remember him arguing with Cesar Milstein in the corridors of the Laboratory of Molecular Biology. In those days, they both used to smoke pipes, and they would have this ferocious argument about how molecules came out of a membrane, whether they were wiggling out or that kind of thing, and how you would test that. And I do remember them with their pipes. Pipe smokers actually argue in a very interesting way, because they have the opportunity, if someone makes an important point, to get their own pipe and decide to relight it and tamp it out, and then take a puff on it, and then decide to now they've got time to think, to decide what to say. And there was this kind of thing going on in the corridor outside. And I think I was deeply impressed by both of them in sense of their intensity and argument, and enthusiasm for the subject, total immersion in the subject. But there are other kind of impacts that I realized I was mentored. Effectively, I was mentored by Cesar Milstein in writing. Now, Cesar Milstein is an Argentinian. He speaks English well, but it's very broken. And you wouldn't actually think that he would write well. But actually, he wrote very well, but it was a very painful process. It was even more painful to write a review with him. I wrote a review once. It's the only paper I ever wrote. And it took months. And I would go in at the beginning of the day, and he was kind of pretty much sort of towards the end of his scientific career. He had all the time in the world. And we would start a paragraph, and perhaps by lunchtime we'd have 2 sentences. And I would say, I was just desperate to get back to doing some experiments. And he would say, no, we haven't quite got this right. It's not quite capturing the essence of this problem. And I remember one occasion we had an argument for about an hour about a particular ending of a sentence, because he wanted to be absolutely precise. And I said, Cesar, actually, you're right. You're right. Let's not have it your way. Absolutely, you're spot on. I see your point. Because I couldn't stand it anymore. He said, no, Greg, actually, do you know, I think you're right. He said, almost right. Almost, I think it's not quite right. He didn't allow me to go on. But what I learned then was it really matters what you put down. Some people just shove something down. And with Cesar, I learned to be much more careful about what I wrote, and try to express it as precisely as possible. I don't think I ever wrote as well as he did, but because actually I didn't have all that time to debate. But he just loved having someone to debate and argue. And I realize that's the other aspect of mentoring. Mentoring's a two-way process. If you have a good mentor, probably you're actually doing a reverse mentor on the other person.
Very good. Thank you.
We've written many papers together, Greg, and I can see now what has happened. I've never really had a scientific mentor. I've been a scientist since ever I can remember, and there's certainly no mentors at school and things like that. But the people who have influenced me most have been those because of their character and the way they've treated other people. I did a postdoc in the States with a very fine man called Bill Jenks. And he was such an upright gentleman and so honest. And is honest about everything. That had a great impression on me. Also, when I finished my postdoc, I went back to the MRC Laboratory of Molecular Biology and shared a small lab with Max Perutz. And he, again, had a huge influence on me by the way he treated everybody. One of the reasons why that lab was so successful was because of the character of Max Perutz. There was no hierarchy there. everybody was treated as an equal. And he would also take so much interest in people. In fact, he taught me how to write. I gave him my first paper that I'd written. It came back to me half scrawled in red with corrections and improvement. And my next paper had about a quarter red, and the third paper he looked at was plain. He'd actually taught me. So I think it really, in my case, I think it stems down to Einstein's saying, aphorism, that the best way to teach is by example, even if it be a warning example. And there are some people around whose behavior's been such, they've been a warning not to act the same way as them.
Very good.
Yeah, well, I think these are really well said. I'd kind of echo some of it. In the talk I gave, I talked about my experience as a postdoc. I should also maybe go back a step and say my experience as a graduate student doing a PhD in chemical engineering. I thought it was the most boring thing in the world. And what was good about when I went to do a postdoc is Judah Folkman, who was my boss, as I mentioned. I mean, what he was doing, I thought was just really exciting. And he also had, this attitude about anything was possible. And he, and I think just seeing that kind of environment was almost kind of as maybe a role model, really changed how I thought about things. And so I'd say that whole experience with him and with that lab really had a powerful effect on me. But it's kind of in some of the ways that you just heard from the last two speakers.
Okay. Professor DeGrado?
Okay, trying to think of some that hasn't been said, but the first was I had two PhD advisors, and really, like, for the first time, I felt like they really believed in me as a scientist, and that was just a huge thing. Let's see. One was very debonair, and that was Tom Kaiser, and he looked after the very big picture, and I had way too many ideas, and so I was always bouncing off the wall with ideas, and he would, sort of go through them and tell me, well, no, but this one's good. He also, once a year, would say, are you going to get bored doing with what you're doing? And I'd look at him and say, yeah, eventually I will. I said, well, get bored now and go learn something different, because you'll never have this opportunity again to learn so many different things. My other advisor was about every detail and rigor. He was the most rigorous person I ever met. His name was Francois Kesdy. And he was always working on our mathematical skills. And he knew how to do every experiment. And so just having that sort of apprenticeship, learning how to do things was wonderful. So from there, I learned, what good mentors were. Throughout the rest of my life, I've always found mentors. Sometimes they're younger than me now, but it's always good to have somebody you look up to and somebody that you can have a dialogue back and forth.
Thank you.
Okay, my experience is completely different from others because I'm female, and it's a long time ago, so very, very rare, the female doing natural sciences. So my high school, it's the prefecture I was grown up, is segregated education, so high school is only for female. My parents did not allow me to go to national university, but it's only female only. Now, I went to Tokyo University for my graduate school, but my PhD supervisor is famous for anomalous scattering, X-ray diffraction, so I'm the crystallographer. But he told me, best thing for women is to get married and stay at home. That's my supervisor told me, but I had to thank him. Because of that, I decided to go to England. That made my career, okay? Because I realized there's no hope I can get a research position in Japan. That was quite true. So I went to England, thanks to England. Because I did postdoc, I got a permanent position in England. I started teaching at Biophysics Department, King's College London. Although English is not my mother tongue, I don't speak good English, but still, there are some people helping me to give lectures. how to make handout. And Maurice Wilkins' actually lecture I continued. He gave me his handout. So there are always people helping you if you want to. So I cannot say I have very good mentors or complete opposite, but I'm always grateful what I am because it's thanks to many people. And I try to be kind to young generation, especially women, if they are really keen to do science or not necessarily science, let's make our world better and understand the wonder of nature together. Thank you.
Thank you so much. We're glad you took the decision you did. So we don't have much time yet, but there's 2 questions up there in the corner. And it's actually, if you don't mind, let's start with a few rows back. I've been promising them. Keep going. Yes.
Okay, thanks for all your great talks, which really inspired me. And I'm Tony from year 1 undergraduate from HQST. So I have a question for Dr. Zhang. So, sorry, I'm nervous. How can we increase the efficiency of finding another nature system or finding another useful protein? Because I searched online that your lab is doing such kind of things, discovery, new nature system. I'm just wondering whether you have some methods to shorten the period of the discovery process, because CRISPR is founded in a bacteria. So I'm curious about how can you improve your efficiency of the whole process?
Yeah, that's a really good question. That's something I think about all the time. So I think, first of all, you have to have a list of questions or a list of needs or a list of problems in mind. So for example, the ability to be able to easily recognize specific DNA sequences, that is a fairly well, precisely defined need. So if you have a list of needs like that, so for example, is there a way to selectively degrade IgE antibodies in the body? Or are there things that can, allow a bacterial cell to be able to colonize an existing microbiome and take residence in that microbiome? So if you have a list of questions like that, then you can start to formulate what a search strategy may be to look for those things in nature. And then once you have that, then I think you can probably set up some type of AI-based system to then go systematically scour through scientific literature and try to find any clue that something may be related to solving the problems that you have in mind. And then once you have that, then you can use structure-based analysis. You can use homology-based analysis. You can use different evolutionary schemes like guild by association or things like that to then start to really look for interesting features in the unknown. So that's how I would go.
Okay, thank you so much. I know there's lots of questions, and I have one more that we're going to ask today. And then tomorrow we start again at 9 o'clock. Make sure you register before, and you'll have other chances to ask questions. But please go ahead with yours, and then we're going to say thank you to all.
My name is Anya. I'm a first-year student at UST, and actually I have a question for Professor Pernilla Wittung-Stapschi. Did I pronounce it correctly? Oh, yes, good. I was wondering, how did you find out that amyloids can act as enzymes?
Can you pass the microphone to Pernilla, who is right there? She's going to be on our panel tomorrow, actually.
But can I answer this one?
Of course.
Yeah. I mean, it was actually not me who was very, very first. There was one publication that said that Alzheimer's amyloids could do chemical catalysis. And when I saw that, I thought maybe this is general to other amyloids too. Because you have this ordered long fiber that could have cavities of kind of active sites. So then we decided to go test it on our amyloids. And then we found more and more of that. So it always is somebody else that has done something that kind of triggers the idea in your head.
All right, thank you so much. Is it a short question? Okay, one more. Right here, this.
Thank you to all the professors for the wonderful presentations. And my question is directed to Professor DeGrado. And I'm Jiang Siqi, and I'm a first-year graduate student from Sun Yat-sen University. And I'm currently interested in drug molecules and protein design. And I'm exploring specific topics in this field, and I find your research particularly inspiring. As I'm still refining my own research focus, I would greatly appreciate any advice you might have for someone starting in this area.
So in protein design, I think the most important thing at this point is to have a good problem to solve. And so choose something that really is important that you can solve by protein design. And whether it's drug delivery, I think is going to be a huge area, catalysts, and also just pharmaceutically active proteins, vaccines. I think, the world's open, but I think you really need to address a very important question. Beyond that, find a lab and that can teach you some of the skills, but the computational part of design is getting easier and easier to pick up. AI has really not only made things better, but they've made it a lot easier. So I hope that answers your question.
Okay, thank you for the valuable advice. As I'm starting to build my foundation, may I ask what specific skills or knowledge areas you would recommend I focus on?
I'd say good computation is important. Good protein chemistry skills are important. And beyond that, finding a good problem, and then you might become an immunologist. You might become a materials scientist. There are just so many different avenues that you can go do. But I'd make sure that when you do that, that you go very deep and you really become an expert in that area.
Hopefully we're going to see you over the next couple days, because one of the important aspects of Molecular Frontiers is to encourage young people to ask the right questions, which I think is what is being said right here. So I just want to thank everybody today. And we look forward to seeing you tomorrow, and register before, and at 9 o'clock we start. So have a good evening, and thank you.